also with you.
Right? Because I see your face and you look familiar to me.
Yeah, you have
Hmm.
done my first class together. We have done the first class together.
What did we do in the first class? Do you remember the topic?
We started cell.
Cell, right?
Yes.
Hmm. Okay, so in the cell chapter, like, did I take your diagnostic test at that time?
Oh, yes, you did.
Okay, so I believe, and do you remember writing anything from that class?
Yes, I.
Yeah, just check and tell me where we were. Because I saw your face and I'm like, okay, I've seen you before. It's been a while, but I've seen your face.
Yeah. Um, we did till difference between prokaryotes and eukaryotics.
Yes, we saw the differences that are there between the prokaryotes and the eukaryotes.
Um,
Hmm.
my exams tomorrow I have bio test.
You
So
have a
for that
biotest.
yeah,
Hmm.
I would like you to revise some topics that's coming for the test that I have doubt in.
Okay, sure.
Um, chapter number 10, cell cycle and cell division. That chapter I want to revise once.
The whole entire chapter.
Yeah, just the important points are, you know.
See, the whole entire cell cycle and cell division chapter, it revolves around how the new cells are formed from the pre-existing cells, right? And the cell cycle, it takes place in two steps. Firstly, there will be the karyokinesis, then followed by the cytokinesis, right? Karyokinesis is the division of the nucleus, and after every karyokinesis, there will be one cytokinesis, which is the division of the cell. But much before this comes another phase, like the whole entire cell cycle, it has been divided into two steps or two cycles or two phases. The first is interphase, and the next is the M phase, wherein actual division takes place. Now, this interphase, it's the preparatory phase. Okay, it's like where all the preparations that are needed for the M phase, those things happen, right? So there is G1 phase, S and G2 phase. These are the phases that you will find in the interphase. All of these are the preparatory phase. So the G1 phase is the one wherein the this thing, the cell organelles and every duplication starts to happen in the G1 phase. The synthesis of DNA takes place in the S phase, that is synthesis phase. And then again, the division of the centriole and everything, that's going to happen in the G2 phase. Okay. So this is like the preparatory phase that will be leading for the M phase. Now this M phase is actually the cell cycle stage. This is where the cell cycle is actually, cell division is actually taking. So the M phase, which you can also call as the mitotic phase, it is the phase where actual cell division takes place, and it is very dramatic phase. It has been divided into two, that is karyokinesis, wherein nuclear division is karyokinesis, followed by cytokinesis, that is cellular division. So remember that every karyokinesis gets followed up by a cytokinesis. Clear?
No.
See, this G1, S and G2, this was like the part of the interphase, and they actually constitute a big chunk of the cell cycle. Whereas the M phase, it has a little roughly this process, right? Now further, the M phase has been divided into prophase, metaphase, anaphase and telophase. Four phases are further of the M phase. Okay, each of them have their significant functions. Now, one more phase is there, which is G0 phase, wherein The cells that are not dividing, that do not wish to divide, they go into this particular stage so as to escape from the division. Clear?
Yes, ma'am.
Now, the mitosis is called as the equational division. Would you like to tell me why?
Because
Why?
the
Say,
cell divides into two identical
daughter cells. Meaning,
cells.
the mother cell, however many chromosomes the mother cell will having, the same exact chromosomes will be present in the daughter cells. And at the end of the mitosis, there is the presence of two daughter cells. So, mitosis, it is the process in which the parent cell divides into two identical daughter cells. Now, these daughter cells, they are identical to each other as well as they are identical to the parent cell. Now remember the reason why it is called as equational division, because the daughter cells and the mother cell, the parent cell, they have the same number of chromosomes. If the mother cell had diploid number of chromosomes, the daughter cell will also be diploid. Clear?
Yes.
Now where does the process of mitosis takes place? So it takes place in the body cells. Okay. Mitosis has another name which is called as somatic cell division. Why somatic? Somatic cells are the body cells. Okay. So when the cells divide, so it's like mostly this mitotic division, it takes place in the somatic cell. Okay. That is why you can call it as somatic cell division. Now, in all those organisms that, like for the multiplication process or whenever there is a need to bring more cells or to replace the old cells, then in that case, mitosis takes place. Clear? Now, let us see the various stages of karyokinesis, which is the actual nuclear division. Now, firstly, there is the prophase, in which what will happen? Prophase is the first phase, and here the chromatin material which is there in the DNA, it will start getting condensed. And condensed means it will start like, it was like a ball of wool, right? Now this ball of wool will open. So untangling of that would happen so that mitotic chromosomes could be formed. Now each chromosome, they appear like a double and it consists of two coiled sister chromatids, like in the prophase. Okay, and they sort of look like the ball of wool. Now at the same time, when the chromosome is sort of untangling, then what happens? Then at the same time, the centrioles, they also duplicate and they move towards the opposite poles. Now remember, these centrioles, they are absent in the case of plants. Apart from that, during the prophase, the initiation of the mitotic spindle also appears. Right? Mitotic spindle starts to form, and at the end of the prophase, there will be no nuclear division, no nuclear membrane present. There will be no cell organelle. Everything will disappear. Clear? After the prophase comes the metaphase. Now, metaphase is the second stage of the mitosis. And here, what happens is that the chromosomes that were formed, like untangling was completed. Now we have chromosomes. Now these chromosomes, they are like thick and they are short. So it's like very easy to study their morphology, meaning body structure. So the best stage to study the morphology of this chromosome is the metaphase. Clear? Now, what happens is that during the metaphase, the chromosomes, they start to align at the equator, meaning at the center of the cell. Okay, and they form a metaphasic plate. Clear? So this bringing of the chromosomes to the equator is called as congression. So congression of the chromosomes take place, as a result of which, if this is the cell and this is its equator, so all the chromosomes will align at the equatorial path, meaning at the center. Clear? And this plate that is formed by the chromosomes aligned, we call it as metaphasic plate. Okay? Now, after that, during this time, the centromere joins the two sister chromatids together. You know that every chromosome is made up of two chromatids which are joined together in the center by centromere, right? So now the centromere, they have a disc-shaped structure which is called as kinetochore. So if the centriole is here, so the centriole sends the spindle fiber, and that spindle fiber comes and attaches to the kinetochore so that it can hold it in place. Clear?
Yes.
So the kinetochores, they are present around the centromere and it forms the attachment for the microtubules. Now, this is like basically the formation of spindle fibers that is taking place. And those spindle fibers, they are going and getting attached onto the kinetochore. Is it clear? See the
This
same
will
exact
be
thing? Huh. See? What were you saying?
so these spindle fibers, they go and get attached to okay, and they keep them in place.
Kinetochore. Right?
Kinetochore.
So where is the spindle fibers coming from? They're coming from the centrioles that went to the opposite poles. So they are sending out the spindle fibers. So from one pole and from another pole, from both the sides, the spindle fibers are coming and they are getting attached onto the point or a disc-shaped structure called as the kinetochore. Clear?
Yes.
So this was metaphase. Now comes the anaphase, which is the next phase. Now remember, like one chromosome was made up of two chromatids, right? And it was joined at the center with the help of centriole. Now during the anaphase, the chromatids, they will split. Imagine like the centromeres, they were holding on to the kinetochore, now they are pulling apart. So they are like splitting the chromatids. So during the anaphase, what happens? The chromatids, they move towards the opposite pole. The centromere which holds the two chromatids together, the centromere splits and the daughter cells, now they become the chromosomes of the future daughters. They each reach the separate pole. Okay. After that, what happens? There is like a fiber, like when they are separating, there's like a fiber formed in between them, which is called as the interzonal fiber. And when they move towards the opposite pole, at that time, like while they are moving, while they are splitting, at that time, you can study the shape of the chromosome very easily. So if someone would say, what is the best stage to study the morphology? So that is metaphase. And the best stage to study the shape of the chromosome is anaphase. Clear? Then comes the last phase of the mitosis, that is telophase. Now during the telophase, what happens? Now the chromosomes, they have reached the opposite pole. Now the mitotic spindle, like the spindle fiber that was there, it will disappear. The chromosomes now again will become entangled. You will not be able to see them properly. And they will become just like how they will lose their identity and become how they were initially. After that, during the telophase, all the cell organelles will come back. They disappeared in the prophase, but now they will come back. And then the nuclear envelope will also come back. So this is how telophase is done. So the whole entire mitosis is like it has been divided into 4 steps: prophase, metaphase, anaphase, and telophase. But before that, there is one more phase, which is the interphase. You can call it as the preparatory phase. Clear? Now, like I said, up until now, what we have studied was karyokinesis, which was division of nucleus. But now nuclei, like the nucleus has divided. Now we'll move on to the cytokinesis. Cytokinesis, cyto means the cell. So the division of the cell is cytokinesis. Now during the cytokinesis, what happens is that the end of the cell division, it is marked by the cytokinesis. Here, the cytoplasm will split. For example, if you see here in this particular picture, this one, the chromosomes went to the opposite poles and then they became Like they lost their identity and they become like this. Something that you can't see where it starts, where it ends. Now, but two nuclei, or they are present in one cell only, isn't it? The cell is one. Now the cell will split from here into two. One and two daughters. So this is what happens during cytokinesis. Clear?
Yes.
Now, let's just say that, in some organisms and in under unique situations, that every pyrokinesis, maybe it doesn't get followed by cytokinesis. So what will it result into?
Sorry?
If the karyokinesis, it is not followed by cytokinesis, what will it result in? Like, what will be the consequence of it?
It
Say,
may not split.
it will not split. No. My question is, karyokinesis is happening. Yeah, you are saying it will not split into cells.
Yeah.
Yeah, that's true. Other than that, what else could you imagine?
The formation of
A multi-nucleated situation will be there because constantly the nuclear division is happening, but the cellular division is not happening. So that will result into one cell containing multiple nuclei, right? And this situation wherein one cell or one cytoplasm has a lot of nuclei present, we call it as syncytium. Okay, a single cell with a lot of nuclei, big number. But always remember that this is not normal. Normal is karyokinesis followed by cytokinesis. Now remember that this cytokinesis, it takes place in the case of animal cell as well as the plants in both the cells. But their mechanism is very different. Do you know the difference between centripetal and centrifugal forces? No. Okay. See, what happens is that when something acts at the center, like from the periphery to the center, like this is centripetal force. Because centripetal means acting at the center, but centrifugal is acting away from the center. Clear? So when we talk about in the case of animals, the cytokinesis happens by the formation of a furrow. For example, see here. If this is a cell like this, okay? This is a cell. Now they are saying that in the case of animals, the cell cytokinesis will happen by furrow formation. So imagine like this. Slowly pinching off towards the center and then they will become two cells. So it's like from the sides, it went into the center. So we say that it is centripetally. So the cytokinesis in the case of animals, it's happening by the formation of furrow, and this furrow is formed centripetally, meaning at the center. Clear?
what is this furrow made of?
Furrow is nothing. It's like deepening.
Okay.
Furrow is deepening. It's not something made like that is made. It's like when the cell is there, it deepens. So this deepened structure is furrow. Okay. And then it pinches off and becomes two daughter cells. Clear?
Yes.
But when the same exact thing, when you have to study this in the plant cell, then it is different. It starts from the middle and then goes at the side. Instead of from the side to the middle, it starts from the middle and goes at the side. So that is centrifugal. Now, just like how in the animal cell, furrow formation was taking place, in the plant cell, the cell plate formation will take place, which is called as phragmoplasm. Okay, so it starts from the middle and then it goes to pinch off from the end like that. And then it becomes two separate cells. So always remember, in the case of animals, it is the furrow formation centripetally. In the case of plants, it is cell plate formation and it is centrifugally. Clear?
Yes.
Any doubts?
No.
Now see, this is like the difference between the cytokinesis that takes place in the case of plant cell and the one that takes place in the case of animal cell. So when the division of the cytoplasm is taking place by the formation of cell plate, that is in the case of plants, then by furrow, then animals. If it is taking place like you can say centrifugally, then that is in the case of plants, and centripetally in the case of animals. Clear?
Yes.
Any doubts in mitosis?
No.
Okay. Now let us see why this mitosis is required. So first of all, growth. See, growth, increase in number and increase in size. This is the function of mitosis. Whenever there is a need to increase the number of cells, whenever there is a need to increase the, you can say, the size of the cell, in all of these cases, mitosis is the best kind of division. Okay? And whenever you want to maintain the size of the cell, you don't want it to become more bigger or smaller, then you would also opt for mitosis. Now, since mitosis is an equational division, meaning the number of chromosomes that is there in the parents is exactly in the offspring. So, the genetic stability or the chromosome number is maintained by mitosis. Now, mitosis is used for repairing of old tissues and cells, and it is used for reproduction in the case of unicellular organism and for healing and generation. So, see, whenever new cells have to be formed, like let's just say our body requires 50 new cells. So, those 50 new cells will be made by mitosis. Clear? After that, there comes meiosis. Now, meiosis is very different from mitosis. It is the common name of the meiosis is called as reductional division. Okay, reductional division. Now, this is a little more elaborate as compared to the mitosis, and it's a little more complicated. It has more number of steps, but all in all, the equal parts. Now, let us see. The whole entire process of meiosis, it takes place into two cycles, meiosis 1 and meiosis 2. Okay. So during this meiosis, what happens? The chromosome number gets half. So in the offspring, like the offsprings, they will have half the number of chromosome as compared to the parent cell. For example, the parent cell has diploid situation, 2n. So in the daughter cell, after the meiosis, it will become 2n by 2. That is n. That is from haploid, it will become diploid. Now, what will happen if the number of cell in the parents or number of chromosomes in the parent is n? So what will be the number of chromosomes in the offspring after meiosis? Tell me. Say, you're right. Be confident. They get halved, right? So n by 2. So whatever is the total number after meiosis is completed, half of the chromosome number will be there amongst 4 daughter cells. At the end of mitosis, 2 daughter cells were being formed. And at the end of meiosis, how many daughter cells will be formed? There will be 4 daughter cells. And 4 of them will be half the number of chromosomes. Okay. Now, where does it happen? See, mostly meiotic division is utilized, or it is very important for gamete formation. The male gametes, the female gametes, they have to be formed, and they get formed because of meiosis. So it happens in the sexually reproducing organisms. Okay. Now the whole entire meiosis, it has been divided into two, that is meiosis 1, meiosis 2. So the subdivisions of the stages will also be like prophase 1, prophase 2, metaphase 1, metaphase 2, like that. So first meiosis 1 takes place, and this is the reductional division. This is where the actual, like the number of chromosome gets half. How? The first phase is prophase. Obviously here also condensation and everything will happen, untangling will happen. But the prophase 1 of meiosis 1 as compared to the mitosis prophase, it's like very, very elaborate and very, very lengthy. Why? Because it has been further subdivided into 5 stages. What are they? Leptotene, zygotene, pachytene, diplotene, and dikinesis. Okay? These are like the subdivisions of prophase 1. Now, during this, like the leptotene situation, the condensation and coiling, all of those things happen. And then you can easily see the chromosome. So this first process is that visibility is increased. Second, there is zygote. Now, those two chromosomes that are homologous, meaning, for example, if this is a chromosome and this is a chromosome, so they are homologous chromosome. Okay, they're like very similar. So those homologous chromosomes, they will come side by side and they will start pairing. Okay. Now this pairing is called as synapsis. And after the synapsis is done, meaning when the chromosomes have paired, now they will be called as bivalent. It has two homologous chromosomes in a pair. After that, during that synapse, like two chromosomes are there, one chromosome, second chromosome. In between them, there is a complex meant to stabilize it, which is called as synaptonemal complex. What is the function of synaptonemal complex? It is responsible for stability, providing stability. Clear?
So when they are pairing, it's known as synapses, and after they have paired, it's known as bi.
Yeah.
Bi.
Synapsis is the process. Bivalent is the structure. After the zygotene gets completed, there comes the pachytene stage, which is the third stage. Now here what happens, for example, see things. This is chromosome number 1, okay? And this is chromosome number 2. Now I am labeling 1, 2, 3, and 4. Now remember that 1 and 2 are sister chromatids, but 1 and 3 are non-sister chromatids. Same way, 1 and 4 is also non-sister chromatids. Clear? So during the pachytene stage, what happens is that there are, you can say, the two chromatids of the same chromosome, they are sister chromatids, and of different chromosomes, they are non-sister chromatids. Now, amongst the non-sister chromatid, meaning this is a chromosome and this is a chromosome, and these two are non-sister to each other, now they will cross over like that, meaning they will exchange their genetic material. And this exchange of genetic material is called as crossing over. So they can ask you, in which of the stages does crossing over take place? So now you know that it takes place in the pachytene of the prophase 1 of meiosis 1. Now, after the crossing over is done, it means that whatever genetic material this had, it's sent there. Whatever this had, it's sent there. So it's like exchange is happening. And this is very important. You know why? Because this crossing over, it leads to evolution. Remember, this is the point that the crossing over leads to evolution. Clear?
So now one will be crossed over with 3, and then 2 will be crossed over with 4, like that.
No, but only one of them. Like let's just say 2 and 3 got crossed over. This is chromosome number 1, like that.
Oh, okay.
This point. This is where the crossing over is taking place. And this place wherein the actual change or the exchange of genetic material is happening, we say that is a recombination nodule. It's like a nodular structure where it is attached. That is called as a recombination nodule. And obviously there is an enzyme that is keeping it alive, keeping it perfectly well, and that is the recombinase enzyme.
Okay.
Now, by the end of pachytene, the recombination between the homologous chromosome gets completed, but they are still attached here. It's like not that they are removed. Whatever they were attached at, That side they will still stay attached. Then comes after the pachytene, there is the diplotin stage. Now, the recombination nodule slowly start to go off, like remove. Okay, but before that, let's see what all happens. First of all, the synaptonemal complex that was there, it will dissolve. Okay, but it will still stay in contact where the crossing over was taking place. It will still be attached. And this structure that is formed because of this crossing over attachment is called as chiasmata. But during the diplotin stage, slowly it is like beginning to slip off. It has to be removed. So now what happens is that the chiasmata, they mark the site where crossing over occur and it went out. Clear? And then after the diplotin gets over, there comes the dikinesis, which is like now moving towards the metaphase, the second phase of the meiosis or meiotic division. Now the chiasmata was there, the recombination nodule, it will fall off, get off. Okay, so that is what happens during dikinesis. Okay.
Actual slip off happens in light guns.
Dikinesis, the last. And now the next step, like this, till dikinesis, the prophase gets completed. Now the metaphase happens wherein the chromosome pairs, right, they start to align again at the equator, but here the bivalent chromosomes are aligning, not the single chromat. Chromosomes are aligning. And in this case, double metaphasic plate is formed. Why double? Because meiosis 1 and meiosis 2, both of them are taking place. So double, you can say metaphasic plate. Like, so here one and then again one. Clear? Like that.
Yeah.
Then after that, there comes the anaphase. Now, ana itself means up. So now during this stage, the chromosomes will start moving towards the pole. Now the intact chromosome or the chromatids, they are going to go to the opposite pole. Okay, and for that, the spindle fibers, they start off shortening. Imagine like you pulling the rope towards yourself. So it will bring that object to you. Same way, that thing is happening in anaphase. Clear? And lastly, there comes the telophase. But here, remember that in anaphase, the chromosome number gets halved. So in the anaphase 1 of meiosis 1, the chromosome number gets halved. Then the last stage of the meiosis 1 is the telophase, which is the final stage. Now the chromosomes will again reach their opposite pole. Okay, they will lose their identity yet again, and they will have the cell division follow. Clear?
Yes.
Any doubts in meiosis?
No, ma'am.
Now, in between the meiosis 1 and meiosis 2, there is one more division that takes place, and that is interkinesis. It's sort of, imagine it like a intermission, like how we have intermission during movies. It's like that only. It's like a break. Now here, everything will be synthesized except for DNA. No DNA synthesis will happen because we don't need any more extra DNA, right? Whatever DNA and chromosomes are present, the meiosis is handling that. Then the meiosis 2, it is also called as the equational division. Okay, equational division just like mitosis. Meaning here the chromosome number will not get half. It got half already in the meiosis like 1. But meiosis 2 is like a normal equational division. Clear? Further, this is also prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, these are all the same exact thing that you have studied. Like what? Like prophase is like it's going to be the compaction is going to happen. The DNA will be visible better. The nuclear envelope, the cell organelle will disappear away. Then all of them will align at the equator and make the equatorial plate or metaphasic plate. Then comes the anaphase. So from that metaphasic plate, they are splitting into two. Like one chromatid went there, one went there. So that is what happens during the anaphase. And last is the telophase. So here the chromatids, they will reach the opposite pole, lose their identity, and then become the new cell. Clear?
Yes, ma'am.
And okay, after this meiosis 2 gets completed, then comes the cytokinesis. Obviously, the cell has to divide. So now the cell divides, leading to the formation of now different kinds of cells that are there. Clear?
Yes.
Now let us see what are the functions or the significance of meiosis. So it's responsible for the formation of gametes, maintenance of the chromosome number, whether it stays diploid, whether it stays haploid, and other than that, introduction of variation. Why? Because if the genetic material was being exchanged, some of this went into this, some of this went into this. So like they're contaminating each other also, and they are exchanging the genetic material as well. So it will be, you can say, very different. Clear?
Yes.
Any doubts?
No, ma'am.
Then the last one, there is, I think this is a little bit extra, but apart from mitosis and meiosis, there is one more kind of cell division, which is called as amitosis or direct cell division. Now, this kind of cell division, it's going to take place in the unicellular organism, like bacteria is there, protozoa is there. Okay, the old disease cells that are there, for that, amitotic division happens. How it happens? Basically, the constituent gets broken into two parts and then the cell is clear.
Yes.
Now tell me any doubts. Like this chapter only is coming, no more else.
There is first chapter. The first chapter is coming, which is the living world and biological classification, cell cycle, and cell division. These 4 chapters are coming.
So with like this, the cell cycle is completed. Now tell me if you have any doubts anywhere.
In the living world, they'll only ask the like main will be the type of
Yeah, they
taxonomic
can ask, they can
categories, right?
ask you what is taxonomy, right? And they can ask you what are the characteristics of living organism. Then apart from that, they can ask you, binomial nomenclature. Very important.
Okay.
Okay, it's like very, very important. Other than that, in that chapter, you can study, the defining features of living organism. Okay, like how do you distinguish between the living and the dead? And this thing, what am I forgetting? Binomial nomenclature, I've already said, right? So this is
Yes.
like the main topic of this particular chapter, that is living world. In
Okay.
cell cycle, I wouldn't be, like it's very uncomfortable to give the important topics because it's everything is important. You can't even miss out a single thing from it. Like it's a cycle, it's a step-wise, series-wise thing. So try to like, do you want me to share these notes with you?
Yes, please.
Yeah, so that you can, once you read it, it will be crystal clear. Trust me. You don't
Okay.
even need to any like read any kind of book or any explanation. No need to even watch the one short videos. This will be more than enough. Trust me. Okay, so I'll share this like notes with you. And remember the sequence of the cell division. It has to be in this way: interphase, prophase, metaphase, anaphase, and telophase. You can make your own mnemonics. I made one. That is, I played my accordion today. I stands for interphase, P stands for prophase, M stands for metaphase, A stands for the anaphase, and the T stands for telophase. So you can make your own mnemonics or you can learn as it is, but just remember that this is the sequence and this is like how it happens. Okay?
Yeah.
Then next chapter is cell. Cell is also coming.
Not
Fundamental
cell.
unit of life.
Biological classifications.
Biological classification. Okay. So
In that like before another teacher, after you, another teacher taught me till um Kingdom Protesta. We finished all of that, so we have. Can you explain Kingdom Fungi?
big kingdom. Yes, I believe. See,
Kingdom Fungi.
kingdom fungi, it has all the members that are heterotrophic. None of the members of the kingdom fungi are autotrophic. They cannot make their own food. Okay, and the kingdom fungi is filled with the fungus, and it's like they are found everywhere. Okay, you name the place and they will be found. So we say very commonly that they are the cosmopolitans occurring everywhere. So they cause a lot of diseases in plants and animals and humans. And other than that, many of them are like eatable also, edible. Now remember that all the members of the kingdom fungi, they are multicellular except for one, that is yeast. The scientific name of yeast is Saccharomyces, and it is the only unicellular fungi. Now, the body of the fungus is made up of hyphae. Okay, hyphae is like a long thread-like structure. And when several hyphae come together, they form a mycelium. So remember, a very famous phrase is that a trough of mycelium is, a trough of hyphae is called as mycelium. Clear? Now this hyphae, it could of, it could be of different types. For example, if the hyphae has multinucleated condition, no cell wall, nothing is present, that is phenocytic hyphae. Okay, the hyphae without any cross wall. But like cross wall means, like for example, if this is the hyphae, okay, nuclei, just like this, nuclei are present. But it's not surrounded by any cell wall. So this is what you call it as phenocytic or cytic. But when you lay walls here like that, now each of them have become a separate cell. Then you will call it as septate hyphae. Septa means a wall. So a wall is formed. So that is why we are calling it as septate hyphae. Clear?
Yes, ma'am.
Now, in terms of their mode of nutrition, they can be saprophytic, like they are particularly heterotrophic. So they can be saprophytic, parasitic, symbiotic, and all of the kinds. Amongst which symbiotic is something you need to understand. Symbiotic association, it's like an association between two organisms that cannot, you can say, imagine their life separate. It's like both of them are going to benefit each other. That is why we are calling it a symbiotic kind. One example is lichen. Lichen is the symbiotic association between a fungus and an algae. Whereas in the case of mycorrhiza, it's like the symbiotic association of fungus with the roots of higher plants, like, the leguminous plants. So lichen and mycorrhiza, these are the symbiotic types of, you can say, organisms. Symbiotic means it's not that lichen is an animal. Lichen is the association. It's like the relationship is called as lichen. The relationship only is called as the mycorrhiza. Clear?
Yes.
Now, in terms of the reproduction, so they can be like reproducing vegetatively, asexually, and sexually. So all the kinds of reproduction are found. Vegetative reproduction is when no sex organs are used. Instead, the body cells are used to make the new cells. This you might have studied in class 10th also, about fragmentation, right? About budding, and then there is fission. Fragmentation is when, upon maturation, the fragments of the hypha or the fungus will be broken, and then each of those fragments, they will develop into new organisms. So that is what we call as regeneration. Moreover, talking about the budding, so a bud is the external outgrowth that is present on top on top of the fungal body. Now this bud, like for example, if this is the fungal body and this is the tiny bud, so it will remain attached until and unless it becomes mature, and then it will fall off. Clear?
Yes.
Now see, after that there comes the fission. The fission, if you remember binary fission, it's just like that. The nucleus will divide and then the cell will. When we talk about asexual reproduction in the fungus, it takes place with the help of asexual spores. Spores like conidiophores, like conidia, sporangiospore, zoospore. These are the different spores that are, like, you can say, the asexual spores. Then comes the sexual reproduction. So it will take place by the sexual spores, obviously. Okay, now let us see. Some sexual spores are oospore, basidiospore, zygospore. These are the names of the different kinds of spores that are there. Okay. Now, when you want to sum up the whole entire sexual cycle in the case of fungus, it takes place in 3 steps: plasmogamy, karyogamy, and meiosis of the zygote. Now, plasmogamy is when the fusion of the protoplasm is happening. Okay, without the fusion of nuclei or anything. It's like the fusion of the protoplasm. After that, there comes karyogamy. Karyogamy is again the formation of the 2 nuclei, right? Like the fusion. Karyogamy is the fusion of the 2 nuclei. Now, after the nuclei, they fuse, they form like a zygote, right? Now, this zygote will undergo meiosis. So, all in all, 3 steps are there, like the plasmogamy, karyogamy, and meiosis of zygote. Clear?
Yes.
Any doubts?
Protoplast is
Cytoplasm.
site. Okay.
Meaning a cell without the cell wall is the protoplasm. Okay.
Yes. Yes.
Now, what happens? Like for example, if this is one hypha, this is another hypha. They come to lay side by side, and then they, like, the business talk actually starts to take place. How? The plasmogamy takes place, meaning the fusion of the plasma, right? So they will fuse, but their nuclei will not fuse. Okay, and then there are many classes of fungus in which immediately the 2 nuclei do not fuse. So it's like one cell having 2 nuclei, but later on they get fused. Clear?
Yes.
Now this time period for which they are not fused and one cell is containing 2 nuclei, we call it as the dikaryophase. Okay, what do we call it as? We call it as the dikaryophase. Clear? Now the kingdom fungi, it has been divided into 4 classes, subdivision: Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. Now each of them have different kinds of mycelia, different kind of spore formation, and fruiting bodies. Remember that the sexual spores, they are made in the fruiting body. Clear?
Yes.
Now, Phycomycetes, this is the first class of the fungus. They're like very, you can say, aquatic kind, right? They can be found, like, other than the water, they are found on the tree stumps and all of those areas, like in wherein the area is actually a little bit dirty, but that is where they grow. Now, remember, they are obligate parasite. Obligate means strictly they are parasitic. Like, they do not have any other mode of nutrition. They are strictly parasitic in nature. Now talking about the septa, which is the wall. So wall of the mycelium in this thing, the Phycomycetes is what? It is the kind wherein, like, you can say, the septa or the cross wall is not present. So the mycelium is aseptate and cenocytic. Cenocytic means the mycelium containing several nuclei. Any doubts? Now, after that, what will happen? When the two nuclei will fuse, a zygote will be formed. And then this zygote will further go into the, you can say, differentiation process. Clear? Now, the fusion of gametes, it is of like various types depending upon the size and everything. For example, if the gametes that are fusing, they are exactly similar in shape and size and everything. So we call it as isogamous situation or isogametes. But when the two gametes that are fusing, when they are not like each other in terms of anything, one is big, one is small, and like that. So they are called as N-isogametes, meaning these two gametes have nothing in common as such. And the last one is the oogamete. Now, this is the one wherein very, you can say, few fungal processes take place. Rather, it is like a, you can say, the more complicated or one of the stages of the different kinds of gamete fusion. Clear?
Yes.
Any doubts up until now?
No, ma'am.
Let us look at the examples. So there is mucor, rhizopus, and albumin. But before we, like the time is about to be over. So before we end this, I would like to discuss with you the most unique class of the kingdom fungi. There is Deuteromycetes. Now, Deuteromycetes, why they are called as fungi imperfecti? We call it as the imperfect fungi. The reason is that in these, you can say, condition, the what happens is like basically all the different kinds of methods of reproduction are known except for sexual reproduction. So you can't make out whether the Deuteromycetes has the sexual reproduction or no, because like it's not, no data has been given as such. Clear? So that is why they are called as imperfect fungi. Right? They are not perfect. They're very weird. Any doubts?
No, ma'am.
Okay, then I hope like one and a half chapter I've done with you. So you're okay. The test that you have to tomorrow, how many marks does it carry?
40 marks.
40 marks.
Yes.
And you're prepared well for your test tomorrow?
Yeah, just I had these few doubts. That's why.
Okay. All right. So see, you keep on revising. I'm sending the notes right now only, so you can revise them. And
Ma'am, do
other,
you mind sending this also?
yeah, I'll send it. Okay. So you keep revising it, keep learning it, reading it several times, and then hopefully your exam will go well. And do let me know